My name is Karen Tybjerg, and I'm associate professor here at CBMR, working on history and philosophy of the concept of diagnosis. And as you may be able to hear, we're moving on to something a little bit different. And this talk is organized by a section of CBMR that deals with the cultural and societal aspect of the metabolic sciences. And in this context, I'm particularly pleased to introduce you to Professor Greg Radick. He was educated at Rutgers and University of Cambridge and is now a professor of history and philosophy of science at the University of Leeds. And here he's worked on a broad aspect, many aspects of the history of biology, such as Darwinism, the sciences of mind and behavior, and perhaps of particular importance to us, the history of eugenics and genetics. Most notably in a book published a couple of years ago called Disputed Inheritance, The Battle Over Mendel and the Future of Biology. And this work was much raved about, not just because it unearthed new parts of history of science, so new historical knowledge, but also because it made a real effort to tell us how we might rethink this history in order to reconceptualize the way we teach genetics and the way we think about it in order to really mirror the kind of complexity of this field that is in evidence in this conference. So now I just want to hand over to Greg, who will be speaking on Mendel's piece as Exemplar or Exception, New Light on from an Old Debate. That was a nice, yeah. Thank you.
Thanks. Well, thanks so much, Karen, for the introduction, and thanks to Julian and the organizers for involving me in the proceedings today. It's a privilege to be on the same platform with Anu, Ingela, and other speakers, and it's a particular joy for me to get to know the CBMR at least a little bit. It's so impressive, not least the relationship with the Medical Museum. That seems to me a model for creative, collaborative relationships between the sciences and the humanities. Now, in that spirit, my talk today is going to be directing our attention back to the scientific past. And I think it's legitimate at a day like Metabolism Day, which is all about bringing the rest of us up to speed with the extraordinary research taking place around us, literally, in genetics and other sciences, to ask, why exactly should we bother with the scientific past when the scientific present and the future that beckons is just so interesting? Case in point, one of the recent headline innovations, at least in the UK, where I'm based, was the approval of the first gene therapy for sickle cell disease. Now, one could, if one was interested, find out about the history behind this innovation. You could find out who the major players are, how the research field emerged and evolved, what difficulties came up and were overcome. And that kind of knowledge is always rewarding in the way that any kind of knowledge is. But you might still nevertheless feel that it's a kind of luxury. You can go there if you want. But if you want to understand the science, which is kind of what we're here for, then you don't really need to go there. And indeed, you could push that further. Yeah, a particular history precipitated out that innovation. But you know what? Had it been other people, had the whats and the whos and the wheres been different, we would still, roundabout now, have a therapy for sickle cell disease. It's a kind of inevitability. And to that extent, to the extent that something looks inevitable, the history behind it kind of doesn't matter. And if you're inclined that way, it's no accident, because the science of genetics actively encourages a kind of incuriosity about its past. And that incuriosity, I think, is encapsulated in images like this. What you're looking at here is the book cover from one of a series of books that came out in around about 1965, 1966. It was the centenary of the lectures and then publication of Mendel's results from the extraordinary crossbreeding work that he did with peas in the garden of his monastery in Brunn, as it was called then, Brno, as it is now. And what do we see there? We see pea tendrils spiraling up into the double helix, right? That image of classical genetics inexorably growing into molecular genetics, right? Just one smooth highway to truth. That, I think, is what that image invites us to think. And that image was stamped in all over again in the most recent Mendel anniversary. 2022 was the bicentenary of Mendel's birth. And there you see, once again, that conjunction of the pea plant and the double helix. Well, what I want to do today is to push back a little bit against that kind of imagery and the incuriosity that it invites. And I want to suggest that there's something to be learned by going back to the very beginnings of genetics in the early 20th century, and in particular to a ferocious debate that broke out over the question of whether it was clarifying or misleading to frame all of our knowledge about inheritance around the sorts of traits that Mendel focused on in his famous 1866 paper, right? Traits, as this image reminds us, which come as cleanly differentiated binaries, right? The seed is either yellow or it's green. It's round or it's wrinkled, right? Purple flower, white flower, obesity, non-obesity, those kinds of binaries. So binary characters which are exclusively caused by genotypic differences. This has been at the center of our knowledge of inheritance from the early days, right? From the earliest. And I want to be looking at that, and I hope along the way to suggest that there is some received wisdom around all of this and the treatment of these characters as exemplary, which is ripe for challenge. I hope that I can also along the way advertise something of the value of the history of science, not just as a record of what happened in the past, but as a repository of possibilities. Some of them realized, some of them not realized, but still worth a look. So the debate I want to tell you about involved a number of people, but principally these two. On the left, William Bateson. On the right, Walter Frank Raphael Weldon, Raphael to his friends. You see them here in around 1890 when they were about 30 years old. They had been friends who were now frenemies, well on their way to becoming proper enemies. By 1900, which is, as you may recall, the year in which Mendel's paper was quote unquote rediscovered, became a talking point throughout European botany and then biology more generally. By 1900, Bateson was based at Cambridge University in England, and he rapidly transformed himself into the greatest champion of Mendel that there ever was. In Bateson's view, Mendel's paper changed everything. It changed everything for biology, and because it changed everything for biology, it was going to change everything for society too. And he turned Cambridge itself into a kind of headquarters of the new Mendelism, as it was known, but it was rebranded using Bateson's coinage as genetics. So that's William Bateson. Weldon, by the early years of the 20th century, was based at Oxford University. And he, like so many other people, took an interest in this paper by Mendel. But although he was impressed by it in limited ways, he rapidly became skeptical. And if we want a kind of visual analogy for thinking about the differences between these two, I think we could do worse than think of a window and a mirror. In Bateson's view, what Mendel had done through the years that he spent purifying his starting breeds, through the meticulousness with which he carried out his experiments, is that he had swept away all of the clutter and the confusion that had blocked the vision of other investigators. And in doing that, he had opened a window onto inheritance at its most basic. Now, that is a view that's very familiar to us because it's the one we learn in our textbooks, which were written by Bateson and his followers. What about Weldon? Well, as I said, Weldon, in a limited way, really admired what Mendel had done. Nevertheless, he concluded overall that Mendel's results mirrored back to him the choices that he had made. The choices he had made about what kind of varieties to include in his study and which to exclude, what kind of traits to focus in on and which to ignore, what sort of conditions to raise his plants under. And indeed, had Mendel made other choices, he would potentially have reached other, maybe even opposite results. And he put these points of view vividly and, as Bateson saw it, infuriatingly in a paper that he published in early 1902 under the heading Mendel's Laws of Alternative Inheritance in Peas. Now, most of the paper is indeed about peas, but there is an interesting passage in the paper which is about humans and indeed about eye color. This is what Weldon has to say. It would almost certainly be possible, by selecting cases of marriage between men and women of appropriate ancestry, to demonstrate for their families a law of dominance of dark over light or of light over dark. Such a law might be as valid for the families of selected ancestry as Mendel's laws are for his peas and for other peas of probably similar ancestral history, but it would fail when applied to dark and light-eyed parents in general, that is, to parents of any ancestry who happen to possess eyes of given color, right? You make your choices, you get your results. The paper also included a rare for its time color photograph of peas that Weldon had collected from expert breeders working in commercial firms at his time. So people who made their living by knowing peas. And if you have a look at the very top row, what you see, there we go, is that in box number 1, we have peas that are unambiguously green. Box number 6, peas that are unambiguously yellow, but in between a spectrum. Right? Weldon's point being that if you don't classify peas committed in advance to Mendel's categories, you don't necessarily see the binaries. And of course, everything follows in a Mendelian analysis from the binaries. And in Weldon's view, this point absolutely generalized. And he made this point in a letter that he sent to his friend, Carl Pearson, inventor of the chi-square test, which Weldon was pretty much the first person to use in that paper. A letter he sent to Pearson in June 1902. What he says is, what he wrote to Pearson was, what all Mendelians do is to take the diagram of frequency and to call a range AB one quote-unquote character, and the range BC the other quote-unquote character of a Mendelian pair. Now, hold on to that image mentally. It's going to matter for a little bit later in the presentation. And I just want to show you one other image from Weldon's letters to Pearson. This comes from about a year later, 1903. This is how people lived before email and social media. They would write letters to each other in which one would dash off, without any fanfare, a solution to the question of how you go from Mendelian units to normal curve distributions. That's what Weldon's done in this letter. Right? He shows, well, it's kind of trivial if you just suppose that there are 5 allelic pairs and suppose that little a gives you 0 increment in stature, big A gives you half a millimeter, and big A gives you a millimeter, you get your normal curve. Not a big deal to a thoughtful, well-informed, mathematically capable biologist in 1903. So Weldon really gets Mendelism. It's not a question that he just didn't get it, or that he was somehow sort of committed to normal curve variation and just couldn't see how Mendelian analysis could handle that. He totally saw that, and yet he was opposed to the idea that Mendelian concepts and patterns should be at the center of the emerging knowledge of heredity. Well, what is it exactly that he did want? I think about it like this. Between 1902 and 1906, Weldon was developing an alternative in which Mendelian patterns are not the baseline generalization, but the special case. So what does he want to emphasize? He wants to emphasize three things. First of all, variation. In Weldon's view, a science of inheritance worth having is true to the variation we actually observe, not a fictionalized, idealized version of it, right? That's what we want to explain, variation as it actually is, and we can't forget that. Second emphasis is what they called ancestry, what we call genetic background, right? It isn't the case that you just inherit a determinant for this or a determinant for that. You inherit a whole suite of determinants from the family line that you belong to. And in Weldon's view, that sometimes can matter to how a bit of chromosome expresses itself in a body. And he regarded that as well understood in 1902, 1903. So variation matters, ancestry matters, and thirdly, environments matter, right? Genetic background forms a kind of internal environment along with everything else going on in a body outside the chromosomes. But then there's also the external environment, the physicochemical environment. So if those are your emphases, you're not going to start everybody off with Mendel's P's and then complicate it. What are you going to start them off with? Well, in a lecture that Weldon gave to a general audience in 1902, he started them off with this organism, which many of you will recognize, Daphnia, the water flea. And he told his audience about experiments done in his lab before he'd gone to Oxford at UCL by his student Ernest Warren, in which it was shown that if you raise Daphnia up in water, as the water gets polluted with their wastes, their spines shrink, until eventually you get Daphnia that are spineless. But if you then take a spineless individual and remove it to clean water, its offspring will be fully spined. Furthermore, the length of the spine in the fully spined Daphnia will be different depending on which variety of Daphnia it comes from. So remember, this is the starting point for a lecture on inheritance. So what does Weldon say to his audience about all of this? He says, now clearly the condition of the spine is not exclusively acquired, and it is not exclusively inherited. It belongs to both categories. A Daphnia of certain ancestry, growing in a solution of certain chemical composition, has a spine of a certain length. We see here, says Weldon, in 1902, the complex way in which the characters of an organism are determined by the interaction of the two sets of factors, the environmental and the ancestral. Now, so far as we know, Weldon never gave this point graphical expression. But in 1909, 3 years after Weldon died, a German biologist called Richard Volterek, also working with Daphnia, published the first norm of reaction curves, which is what he called them. And for those of you who maybe aren't familiar with these diagrams, right, on the y-axis, we have phenotypic values. So here it's head length rather than spine length. On the x-axis, you get environmental values, here quantity of nutrient rather than quality of water. And each of the curves, A, B, and C, represent different varieties of Daphnia, and we've now represented as different genotypes. And what you see at a glance with a norm of reaction diagram is that phenotypes are the variable upshots of particular genotypes in particular environments. That's unmissable from a diagram like this. By contrast, think about the visual emblem of the Mendelism that we're familiar with, right? The Punnett square. Punnett squares with their blockily binary characters in which nothing matters except the allelic combinations, right? It's entirely possible to be educated within Punnett square genetics and not take into your bones the importance of environments and the variability that can be produced. Now, why is it that we ended up with a science of inheritance anchored in Punnett squares rather than norm of reaction curves? Well, there might have been good reasons for it, but if there were good reasons, that wasn't what decided it. What decided it was Weldon's death. In the spring of 1906, at the age of 46, a guy who at the drop of a hat would go on 20-mile cycle rides, got pneumonia and died, with the book that he was working on setting out his alternative perspective, unfinished and unpublished. And the published Weldon is nothing like as extensive or interesting as the unpublished Weldon. And from that point onward, we have what we're familiar with, right? If you think about a body of knowledge as having a center and a periphery, this is what we get, right? We start out, we imprint on, we anchor on Punnett squares. And no one's in denial about the complexities, but they're not what people remember. People who don't go on to become professional scientists, right? What do they remember? They remember dominance, recessiveness, blue eyes, brown eyes. But when you're aware of Weldon and you're aware of just how close run a thing it is that we ended up with the kind of genetic knowledge that we have, you can't help but wonder, well, could it have been different? Could we have had this alternatively organized, this flipped knowledge of inheritance in which what's central is what became peripheral and vice versa? Now, I think that would be a kind of timelessly interesting question, right? But I think it's especially timely now. And I think it's especially timely now because of developments in 3 areas: in genetics, in science education, and in cognitive science. Let me just take you through those briefly. First, on genetics, and you'll all be much more familiar with this than me, nevertheless. In November of last year, Bruce Gelb, as president of the American Society of Human Genetics, made the theme of his presidential address incomplete penetrance and variable expressivity. In Bruce's view, it was really important for the human genetics community, both the researchers and the genetics counselors, to come to grips in a much more thoroughgoing way with what it meant in the 2020s, to be working in a world where the cheapness and ubiquity of sequencing was sending penetrance percentages down, right? Because we're no longer just dealing with families in which a disease is conspicuous. We're now casting the net much more widely. So incomplete penetrance is seen to be a much bigger problem than it was before, and likewise variable expressivity. So that was the theme of Bruce's address. And it doesn't take more than a little bit of a dipping of the toe into the literature to see where he was coming from, right? 10 years before Bruce gave his address, geneticists at Mount Sinai launched what they called the Resilience Project. As far as I know, the first attempt to sequence the genomes of apparently healthy people, looking for what the textbook said were disease genes. And they found them. At the first attempt of anybody to do this in a systematic way, they found 3 individuals who had a double dose of what we think of as the cystic fibrosis mutations, but they were fine. More recently, 2022 article, A Spectrum of Recessiveness Among Mendelian Disease Variants in UK Biobank. It was there for the finding once you start looking for it. 2023, Exploring the Complex Spectrum of Dominance and Recessiveness in Genetic Cardiomyopathies. Here's a little quotation from that article. The considerable genetic complexity of Mendelian diseases is increasingly recognized, with many conditions characterized by substantial genetic heterogeneity, incomplete penetrance, variable expressivity, and sizable proportions of patients where causal variants are not identified. While Mendelian disease genes are typically categorized as being associated with either dominant or recessive inheritance, this is probably an oversimplification of the actual relationship between genotypes and clinical phenotype. In particular, strict Mendelian definitions of dominance, right, where monoallelic and biallelic genotypes have similar phenotypes, and recessiveness, where heterozygous variants have no phenotypic effect, are unlikely to reflect the reality of human clinical genetics for most Mendelian diseases. Going back to the example I gave you at the start, the sickle cell therapy, as some of you at least will appreciate, how does this work? This works by exploiting something that was observed about the population. There were people who were found to have a double dose of the sickle cell mutations, but they were fine. And they were fine because fetal hemoglobin had never been turned off in them. Right? So again, you can have the gene for, but not have the condition, something that we're familiar with, but these things bring home all the more forcefully. And the therapy works not by fixing the mutation that affects hemoglobin, but by taking the molecular brake off the production of fetal hemoglobin. That was 2023. 2024, the biological anthropologist Benedict Hallgrimsson at the University of Calgary gave a presentation in which he put up this diagram. It expresses Benedict's view that Mendelian disorders ought to be seen as extreme expressions of developmental potentials with polygenic bases. Now, if you're looking at that and thinking, I've seen that before, you're right. It looks rather like the diagram that Weldon put in that letter in 1902. And when I pointed this out to Benedict, he was startled. And I pointed out to you by way of underscoring the point that maybe now is a timely moment to rethink the present in the light of this until recently forgotten past. And then even just within a few weeks ago, in the news in the UK and in the USA, news about a paper in which a family in Oklahoma was found where everybody inherits a mutation, which in almost everybody, it leads to early onset Alzheimer's, except for the one guy who also had the mutation but was fine into his late 70s. Last example, patterns of X-linked inheritance, a new approach for the genome era, right, where we read first line of the abstract. The concepts of X-linked, quote unquote, dominant and recessive inheritance originated long before dosage compensation for X chromosome genes was understood, but now have no scientific basis. So developments out of genetics. Secondly, developments in science education. And here, some of the classiest work has been done by Brian Donovan, based at the University of Colorado at Boulder. What Brian showed some years ago was that when teachers take their middle school or high school students in the kind of classical way from peas to humans via classical Mendelian recessive disorders like sickle cell disease, affecting people of Black African ancestry, cystic fibrosis affecting people of white European ancestry, Tay-Sachs disease affecting people of Ashkenazi Jewish ancestry. When they do that, the students come away with their Punnett square solving skills honed, but they also come away with an exaggerated notion of the homogeneity of the human races. They come away with exaggerated pessimism about the ability to close gaps in educational attainment between the races. They even come away less willing to socialize across racial lines. One of the teachers who was involved with the study said it was as if the students came away from the lessons convinced that people belonging to different races were different kinds of animal. In the most recent study that Brian's been involved with, which was published in Science last year, what he showed was that wherever you drop in standard issue elementary Mendelism within a curriculum, you get an uptick in racist attitudes among the students. So there's something that needs thinking and talking about there. Finally, from cognitive science, Marina Dubova and her colleagues have a paper that's either out now in PNAS or is coming out with the wonderful title, Is Occam's Razor Losing Its Edge? New Perspectives on the Principle of Model Parsimony. And it takes up what you might have thought doesn't really need thinking about, which is the question of whether it's always better to start simple. And the answer turns out to be one that you can assess empirically. And it turns out that the answer, perhaps unsurprisingly, is it depends. If you can already be confident that what you're studying is something whose complexities you've already grasped, that is to say, you can be confident that you're not going to be excluding what turns out to be important relationships, then yeah, going maximally simple can be a good research strategy. But if you can't be confident in that way, you're better off with models that capture some of the rich complexity in the data. And only when you've done that to generate simpler models, but based on that background complexity. And this is a theme which has been picked up more recently by the science writer Philip Ball, some of you may have heard of, in a commentary he published for Cell Systems, Should Biology Put Complexity First? You can guess his answer. Now, in the light of all of that, you might think, surely the case for a rethinking is compelling. And yet you can think that and still not know where to go. I mentioned that 2022 was a Mendel bicentenary. In that year, the human geneticist Aoife McLissett published a kind of paean to Mendel, in which she wrote the following, and I'll fill in the X's in just a moment. She wrote, genetics is incredibly complex. We now know that most traits, physical, biochemical, behavioral, are influenced by many different genetic variants, individually of small effect, acting in combination with the environment and stochastic processes during development. Even identical twins who share all their genes are not actually identical. And she went on from there to express her worries that in spite of her best intentions, her students were picking up a kind of determinism about genes and a kind of essentialism about people that she absolutely didn't intend, but which seemed to be encoded in the curriculum. And yet she began, I could not imagine trying to teach genetics without starting with Mendel. That's what it's like to grow up in an intellectual monoculture, right? That you just can't think, what else would you do? It seems obligatory. In order to unthink all of that, it helps to go historical. And I'm pleased to say that I was able to think of a way of teaching genetics without Mendel. And it's because I was spending time with Weldon, who didn't have to unthink Mendelism. And so I asked myself, well, did this debate between Weldon and Bateson really matter? What if Weldon had lived long enough to publish that book and it had some effect in the world? Would it have made an impact? Well, of course, I can't go back and give him more life, but it occurred to me that I could write a curriculum in introductory genetics, which was as if it came from the Weldonian past that never was. And I thought, well, I'm not going to start them off with Mendel's peas and complicate it, but maybe I could start them off with something like the role that genes play in the condition of a human heart. That's going to be a multifactorial story. It's not going to be a difficult story, but it's going to be properly multifactorial. And it will bring in development and environments and age and sex and stress levels and all kinds of things that I want to have in the causal mix. So let's suppose I could start the students there. And furthermore, throughout the course, I treated interaction not as this kind of cognitive luxury item that I mention and they can forget about it afterwards, but as what the course is about. All the interactions which are pervasive and bring about variability, so that when they meet Mendelian patterns later in the course, they meet them as an interesting special case, as what you get when you strip out all of that variability that ordinarily obtains. So suppose I could do that. What would those students be like? In particular, what would they be like when it comes to questions about determinism? Right, that over-exaggerated notion of the power that genes have over bodies and minds. Well, to my amazement, we got a grant to run the experiment, got a brilliant postdoc, Annie Jameson, who had been a developmental biologist and became a historian of science. So it was sort of perfect to do the work, to recruit the students, to assess their attitudes before and after teaching to genetic determinism, as we did with students taking the ordinary Mendelian course. And then we published the results, first informally in Nature in 2016, and more formally in Science and Education in 2017. And what we found was that students coming out of the ordinary Start with Mendel course were as deterministic about genes at the end of teaching as they were at the start, maybe slightly more so. Whereas students coming out of the Weldonian experimental course were less deterministic about genes at the end of teaching. And at least among the genetics educators I spent time with, that's a win. Right? The more you learn about genetics, the less confident you should be that you can just read phenotype off from genotype. And right now I'm collaborating with Brian Donovan and also Michelle Smith at Cornell to take what we did at Leeds about 10 years ago to the next level of rigor. But also looking at the potential psychological mechanisms underpinning, looking at the potential connections between categorical thinking, stereotyping, and the social prejudice that it perpetuates, and what psychologists call growth versus fixed mindset. A fixed mindset being one which says, I can't change, you can't change, the world just is what it is. A growth mindset being the opposite of that. So when you take that all in all, when you take together the reconstructed history of the debate between Bateson and Weldon, the new developments in genetics, in cognitive science and science education, and the results of this Leeds Pedagogies project in teaching genetics differently, I think that we're now better off anchoring students not in Mendel's peas, but in Weldon's. And I'm going to finish off with two final thoughts. One is that as part of the new sort of teaching, it's worth asking ourselves, might history be of help here, not just in generating the thoughts, but in helping the pedagogy along? I don't know about you, but in my biology textbook, I learned the story of Mendel publishing his paper, getting no response, and then in 1900, as my biology textbook put it, biology caught up with Mendel. Now, if you learn that biology caught up with Mendel in 1900, you have zero motivation for questioning the wisdom of beginning your own genetics education anchored in Mendel's patterns, right? That just seems to be where biology was heading all along. But if you learn instead, apropos of the book that Karen kindly adverted to in her opening remarks, that the history of genetics, much more importantly, begins with a debate between two well-informed people who disagreed about what should and shouldn't be at the center of biology, and that debate could have gone otherwise. You end up being much more open-minded about how you might proceed from there in the study of genetics. I'm going to end by introducing you to a Leeds colleague of mine. This is Tom Bennett. Like you, he heard me lecture on these themes some years back, and he told me later he had two reactions: surely not, and also, how dare you? But bless him, he spent the next 5 years thinking about it. And in those 5 years, he went from being a postdoc in genomics at Leeds to being a member of staff. And he became a member of staff who was asked to teach introductory genetics. And he sent me an email in which he introduced himself, explained he'd been in the audience that day, spent 5 years thinking about my work. And once he was actually faced with the challenge of standing up in front of students and teaching them genetics in the 2020s, he had a look at the hand-me-down materials and thought, I can't. And so he wrote to me to ask, how do we work together to try to figure out how to make a genetics curriculum fit for the 2020s? So you can join in that work too, and I'll end there. Thank you very much.
Thank you, Greg. And now I'll open the floor to questions. Yeah, one here.
Ken Arnold, CBMR. I'm from Medical Museum. Thank you so much, Greg. Such a fantastic talk. I'm going to be greedy and ask you two very quick questions, and you can either answer one or both, or just move on. One is historical, which is, does simple almost always win out? So is it almost always better to... Well, maybe I shouldn't draw any conclusions, but is that your sense that in disputes about ideas that simple often historically ends up being the champion? And the other one is to take you back almost to your last slide, a more pragmatic question, which is after the education, after the teaching, how can we get back to this? You had this fantastic phrase of the repository of possibilities. We're sitting in a room of 300 scientists past much of their education. How can this repository of possibilities be opened up to our extremely busy scientific colleagues?
Super questions. On the first one, for sure, simplicity helps. But if one looks at the record, what one sees is that sometimes simplicity isn't enough. And sometimes the more complex wins out. Two examples: the humoral theory of disease. Anyone? Right? That was pretty simple, but it died a death eventually, replaced by something more complex. Likewise, if you compare an Aristotelian analysis of motion and a Newtonian analysis of motion, the Newtonian one swept the boards pretty quickly once it was adopted. And I console myself with that when people push back against what I present by saying, well, sure, the Daphnia case is interesting, but it's so much more complex. I said, well, imagine if people said that about, gosh, this Newtonian analysis is really interesting. But, the Aristotelian analysis in terms of Earth heading toward earthy places, that's going to stick forever. Now, for your second question, a colleague of ours called Ha-Seok Chang, a philosopher of science at Cambridge, has a proposal for what he calls history and philosophy of science as complementary science, complementary with an E. And what he means by that is that, at least for certain purposes, it can be valuable to have scientists and historians and philosophers of science in harness, but not necessarily in the same lab, down the hall. So scientists ought to be allowed for their purposes to shut down certain kinds of question and get on with the job. But they shouldn't be allowed to do that in a permanent way. And so by ensuring that there are historians and philosophers of science down the hall who will meet at tea time or whatever it might be, they're kept in contact with all of the questions that they prefer to have shut down or maybe didn't realize had been shut down. And I think that's a practical proposal for keeping the possibilities, which you only become aware of if you have the luxury of devoting your life to scholarship in the scientific past, lively for scientists themselves.
Thank you. More questions? Yep, up there.
Hi, thank you for a very nice and engaging talk. I'm curious, like, what your thoughts are on how this Mendelian mindset affects public health, for instance. I'm also thinking, like, okay, around cancer, like, how much can genetics explain cancer? Just as an example, like, maybe you have this, like, BRCA1 mutation, so you think, oh, I should be screened for breast cancer. But how much of these other factors actually play into that? Because of course, just the screening, you can get, you might have to go through an unnecessary biopsy. You might even have unnecessary cancer treatment, which is completely awful. And that's just like one example, but I'm thinking how this kind of more simplistic mindset of Mendelian genetics can affect actually public health, informed consent and decisions.
It's a great and important question, and in my experience, the cancer example, it brings it out vividly. In the UK, perhaps in Denmark as well, the NHS has made an effort to get the word out that unless you have a history of breast cancer in your family, then getting a test for the BRCA1 mutation isn't something that you should just run out and want to do. Why? Because if there is a family history and you have the mutation, the risk is around 80%. But if you don't have the family history and you have the mutation, the risk is around 20%. Well, what does that mean? It means, apropos of the things I've been talking about, that ancestry, in Weldon's talk, genetic background, matters to how a mutation has an effect on a body. So my impression is that the NHS really wants to get that message out there, but that they're really struggling. And they're really struggling, I think, in part because they're working with a population that have been educated within, and then the message has been reinforced, the gene for notion, right? That is, so if you're going to get the gene for if you have the gene for breast cancer, then you should be worried that you're going to get breast cancer. And I have a conversation scheduled for Friday with a genetics counselor working within the NHS who contacted me having read my book because he feels that his options are constrained because of the Mendelism baked in to the way that the NHS deals with these kinds of topics, and relatedly, that he wants a set of resources that would help him in the clinical encounter to deal more capably with the uncertainty that often comes with sequence data. So it does seem to be a really important problem. At least some genetics counselors are persuaded that Mendelism is part of the problem, and so getting the Mendelism out might be part of the solution.
Yep. Well, then if there are no more questions, then I'll take the chance to ask John just one final question, and that is, I got the feeling that the curriculum you taught, you started off with the heart, so you really started off doing it purely biologically, just with another centerpiece. What's your thinking on actually drawing in the history into the teaching? So also teaching a little bit of Weldon and this debate, and do you think that would make a difference?
Well, this is what. Thank you for the question. It's what I meant to be gesturing towards at the end about how bringing the history into the curriculum might actually help. So when we did the teaching experiment at Leeds, we deliberately left the history of science out as a kind of confounding variable. We wanted to see what effect it would have to teach this curriculum as if it dropped from the skies, just like the alternative curriculum. But in all other respects, that isn't remotely how I think about what history of science can do for scientific education. And interestingly, it was left to scientific educators who took up the work, who began to show me that actually it helps when you bring into the classroom, not least in the name of inclusivity, the fact that there was a debate that almost no one but historians of science had heard of. It was a debate that unfolded in the area of eugenics, which wasn't a theme that I emphasized here, but it's pretty important for understanding why it is that when Bateson and his allies go out into the wider world and emphasize the value of Mendelian knowledge, they emphasize that if it has a social message, the social message is, as Reginald Punnett put it in his book Mendelism, the creature is born, not made. Right? That notion that I'm a geneticist and you can't handle my truth, that starts with Bateson and his allies in the heyday of eugenics. So the science educator said that if you bring that into the classroom, students that are otherwise alienated from genetics, not least because they feel that their kind of people have been at the hard end of genetics in the past, feel like they have a way in. And some of the most encouraging responses I've had from the teaching I've done have been from students who said, I used to hate genetics, couldn't find a way in, couldn't wait to not do it. But seeing it as a human product in this way, as part of history, and as part of a history that we can have some agency over. We can have some agency over the stories we tell about it. And when we tell the right kinds of stories, it encourages us to have more agency over the science and indeed over ourselves. That all of that changed her attitude to genetics education. So not every student's going to need that, but there might be some students that we're missing because of teaching it just the one way.
Wonderful. Thank you. And thank you for telling us that history is not a confounding variable and it's not a luxury cognitive item. This is a luxury, not cognitive item. But thank you very much. And let's thank Greg.
Thanks very much. Thank you.